Okay, having looked at some of the basic anatomical features of the cell and understanding a little bit about the cell in terms of, um, glial cells and neurons and understanding their distribution in the brain, now we're going to take a look at how the cell actually communicates, how nerve cells communicate. And again, this lecture is going to be about the action potential. And understanding the basics of this is going to be especially crucial for understanding a lot of the work that goes on in behavioral neuroscience that has to do with drugs, has to do with hormones, has to do with different chemicals, biochemistry of the body, biochemistry of the brain. So to understand the biology of behavior, you have to have a foundation in this. So let's begin this examination. A little cartoon here. I have two neurotransmitters there, or two neurons there, and one says to the other, excited to meet you. And again, this is in response to the question, what did one neuron say to the other? So the excitation of the central nervous system is really dependent upon what is happening in these neurons in terms of the action potential. So it's important that we understand some of the basic features. So this is an important figure that you see here that really outlines some of the things that we're going to be talking about as we go along. The graph that you see there is showing a number of different things, and let me try to outline those things for you. First of all, each neuron has what is called a resting potential, which generally is in the neighborhood of about minus 70 millivolts. That is the difference that you see between the inside and the outside of the neuron. It's slightly negative, the neuron is in its resting state. When an action potential occurs, a neuron is sending information down an axon, and it's doing it as a function of both electrical changes as well as changes in what we call the permeability of the membrane. So there are changes that occur in the permeability of the membrane to sodium and potassium. There's changes that are occurring in terms of polarity, the charge of the neuron. The neuron is, instead of slightly negative, is going to depolarize and become more positive. So let's take a look at what happens here when there's an actual charge, an action potential that occurs when the neuron reaches a threshold point where it can fire. You could see that it is depolarizing and becoming less negative and more positive. And again, this is time along this axis that you see here. And you can see that once it reaches this point of about plus 30 millivolts, then it's going to go down to a more negative state, and then eventually is going to become back to its resting state of minus 70 millivolts. When you take a look at what is happening in terms of these very important protein channels, sodium and potassium channels, what is happening in terms of sodium is that you see in this yellow phase right here, sodium channels are opening up. And again, that's allowing the transfer of sodium into the cell. And you have those channels that are opening up again here in the green, and that's what's going to assist the firing of the neuron. Then those channels are going to close down, and sodium is not going to be allowed to go through them. So they close down, but simultaneous with that, during this blue phase that you see right here, potassium channels are opening up. And those, and then ultimately are going to be closing down. So that's going to assist the neuron charge now going from this positive state back to its resting state that you see right here. So again, two fundamental changes in the permeability of the membrane to sodium and potassium, and changes that are occurring in polarity, meaning the charge of the neuron. So each neuron really can be considered to be like a battery. Here you have a typical battery. Here's the positive end that you see right here. Here's the negative end that you see right here. Each neuron is really like a battery. So if you take a look at what is happening in terms of the voltage here, again, in this resting state, you have this slightly negative state of the neuron. You have a lot of sodium that's on the outside that you see here, mostly potassium that's on the inside. And at the start of the impulse that you see right here, you're getting some sodium now that's starting to come into the cell. And then once that neuron fires, again, here's the end of the impulse that you see right here. You have potassium now that's starting to go out of the cell. And again, you're going to have a restoring of that resting potential. So you can really think of, each neuron as being like a small battery. So here are some key concepts. We're going to talk about firing. We're going to talk about changes in the membrane permeability. We're going to talk about changes in axon polarity, refractory periods. If you have these 4 things down in terms of a basic understanding of them, then it's really going to help you a lot in terms of understanding the functioning of the action potential, and that in turn is going to help you to understand some of the things that we're going to be talking about in terms of the biology of behavior, especially as it relates to the impact of drugs. So one of the analogies that I try to use is that an action potential is very much like a toilet that's flushed in your average household. And I'm going to be using that analogy to explain different components of this of the action potentials. And I think that this visual will help you as we go along. So first thing, the all or none property, when you flush a toilet, you push this lever that you see right here, and that's going to allow this the water to escape, to go down into the toilet, and then eventually out into your sewage system. Pushing that lever harder or faster doesn't produce a bigger flush. That's what we call the all or none property. Pushing a neuron past its threshold doesn't increase the size of the action potential, stays the same. If it's just barely gets up to threshold, or if it's way beyond threshold, it's not going to cause that neuron to fire any faster. It's not going to cause that neuron in terms of the action potential, to last any longer. So this is what we call the all or none property. The refractory phase, again, use our toilet flush analogy. When the tank is full, the toilet, until it's full, it's not going to be able to be flushed again. But what you see in terms of the action potential is very similar. These refractory phases, until sodium channels recover, the neuron can't produce another action potential. So you have this refractory period where nothing can really happen, even if you have a stimulus that's a very, very strong stimulus. Now, we're going to talk about 2 phases of that refractory period, but for the most part, just consider the fact that this refractory phase is a time in which the neuron can't be fired. Another component of this, of course, is the direction of the action potential. Where is the water going here in terms of our toilet flush? Well, it's going in one direction. And again, it's going down and ultimately out through the bottom of the toilet mechanism. So the direction is always in one way, and the same holds true for an action potential. Action potential always travels in one direction. It always travels down the axon. It does not come back again from the axon terminals back up to the axon to the soma of the cell. It just travels in one direction. So here is again our motor nerve that you see here in this diagram. There's an electrical message that's being transmitted down this axon from this axon hillock area. It's being transmitted down the axon to this presynaptic terminal area, and it's just traveling in this direction that you see here. But one of the things that we know is that it is being regenerated at different points, namely at these little gaps that are called nodes of Ranvier. The speed of a neural impulse ranges anywhere from about 1 meter per second to 100 meters per second, but it's always traveling in this direction, and it's being regenerated at these spots here that are called the nodes of Ranvier. And I'll get into the mechanism of that in just a few moments. So our resting potential, the membrane of a neuron has what we call an electrical gradient. And again, here's our membrane that you see here. It's the difference in the electrical charge between the inside and the outside of the cell. And you can see inside the cell is slightly negative, and the outside of the cell is positive. And in terms of our resting state, then of a cell that is prior to the time in which it fires, that resting potential is slightly negative. Again, if you were recording, doing a recording on the inside of the cell and on the outside of the cell, that is, what you would obtain. You would obtain a difference of about -70 millivolts. So again, here's our recording from inside a neuron, and it's about -70 millivolts. Okay. That's our resting state. The membrane maintains this polarization or difference in the electrical charge of these two locations. So the inside of the membrane is slightly negative with respect to the outside. And again, it's about -70 millivolts. So talked a little bit about permeability of the membrane by way of these protein-based channels. The membrane is what we call selectively permeable. It's going to allow changes to occur in terms of sodium and potassium, calcium chloride as well. But when the membrane is at rest, the sodium channels are closed, potassium channels are partially closed. There's going to be, some slow passage that's going to be occurring of these charged ions. Here's what this all looks like. Again, kind of an artist's rendition of what these membrane channels look like. Consider them to be like a screen door. When that channel opens up, it's going to permit, in this case, it's going to permit potassium to leave the inside of the cell. When they're closed down, as we see here and here and here, again, this is the membrane of a neuron. These are these channels. Here you can see this is a potassium channel that's closed. Here is a sodium channel that's open. Sodium is going to be allowed to come into the cell. Here is a sodium channel that is closed that you see right here, so no sodium can come in. So again, when a channel opens up, it's going to permit its respective ion by way of that active transport system. It's going to allow that charged ion to either go inside or go outside of the cell, depending upon the charge. So again, this is what these membrane channels look like in the membrane of the neuron. So the sodium-potassium pump is a very important concept, and here's our pump that you see right here that's going to be allowing 3 sodium ions out of the cell. Again, this is during the resting state. It's going to be drawing 2 potassium ions into the cell. And what that is going to do is it's going to allow this electrical gradient to occur. So again, here's our sodium ions being pumped out. Here's our potassium ions being pumped in that you see right here. That electrical gradient and the concentration gradient in terms of those ions, that's going to pull sodium into the cell. The electrical gradient is going to pull potassium ions into the cell. So again, this sodium pump concept is very important for understanding, what the resting state looks like in the case of a neuron. So again, here's our axon that you see here. This is showing the distribution of ions. Again, this is in the resting state. This is primarily sodium that's on the outside, a little bit of potassium, not much. Mostly potassium on the inside, a little bit of sodium, but not much. So again, the movement of ions that you see here in terms of our sodium-potassium pump. Again, sodium being pumped out, sodium being pumped in. Sodium leaves the cell because of the concentration gradient, and potassium is entering the cell because of the electrical gradient. So sodium ions and potassium ions are crucial during this resting state that's going to maintain this minus 70 millivolt charge that we see in the cell. So the concepts of hyperpolarization and depolarization are important. In the resting state, the cell, once again, is slightly negative on the inside. So you have this resting potential that's stable. When you get hyperpolarization, that's the increasing polarization, or the difference, if you will, between the electrical charge of 2 places. So in this hyperpolarized state then, where it's mostly negative on the inside, positive on the outside, and that negative state is extremely important in terms of the resting state of the neuron. So depolarization is what happens when that neuron fires. You're getting a change from being very negative, that resting potential minus 70 volts, to being less negative, then ultimately to being positive. That's what we mean by depolarization. So the threshold of excitement, that's when the neuron charge reaches a certain level, and that neuron charge, the stimulation that's produced by an adjacent neuron is going to cause that charge to depolarize and ultimately to result in a firing of that neuron. So again, here's a good visual. This is an action potential, and we're getting this rapid depolarization that is occurring. Here's our threshold of minus 55 millivolts. Once we get over that minus 55 millivolt threshold, it's going to cause depolarization to occur. The action potential, the firing of the neuron. Then what you get is repolarization, again going down from this positive state to a more negative state. And you have this refractory period we'll talk about in a little bit more detail, and then this back to this resting state of minus 70 millivolts. Stimulation of the neuron past the threshold of excitation is going to cause that neuron to fire. It's going to cause this action potential to occur. And again, this is all occurring very, very rapidly that you see here. So here's another important figure. This is showing the sodium and potassium ions during an action potential. Again, here's, take a look at the top portion of this. This is showing what's happening in terms of the electrical potential. Here's the resting state. Now it's becoming less negative and more positive. The neuron now fires, and now it's going to become less positive and more negative. All of this is happening very, very rapidly. Okay. Take a look at what is happening in terms of the rate of entry of sodium and the rate of exit of potassium. Red is sodium, and again, associate this with what is happening here. You're having a change in terms of the rate of entry of sodium. You're getting a dramatic increase in terms of that entry. Then sodium is going down in terms of its rate of entry, and associated with that now in the blue is this increase that is occurring in terms of the rate of exit of potassium from the neuron. And again, this is going to allow that neuron to get back to its resting state. So after an action potential actually occurs, sodium channels are going to quickly close down. The neuron is going to return to its resting state. And again, that's by virtue of the opening up of potassium channels. That sodium-potassium pump that we talked about earlier, that's going to restore the original distribution of ions. So again, what we have now is that slightly negative state, that minus 70 millivolt negative state. So the all or none law says, that doesn't matter how the amplitude and velocity of an action potential really are independent of that stimulus that initiates, whether it's one that just barely gets to threshold or it's one that's super threshold that goes way, way beyond that minus 55 millivolt. It doesn't matter. The action potentials that are going to occur are going to be equal in intensity and speed in any given neuron. So again, you go back to our toilet flush, analogy that, it doesn't matter how hard or how fast you press down on that flushing mechanism, you're always going to get the same result. That is, the flow of the water is going to be identical. Same holds true in terms of an action potential. We have these refractory periods that are important to understand. After an action potential, what is going to have is you get this firing that occurs of the neuron, and during this phase that we see right here, this colored area that you see right here, this is called the absolute refractory period. And that means that the neuron can't be fired, even if it's a way, way above threshold stimulation. It's not going to, the action potential simply cannot occur again during this period of time. A little bit later on, however, is what is called the relative refractory period. That's in the yellow. During that second part of the refractory period, then a stronger stimulus, one that's above threshold, way above threshold, can trigger an action potential. Okay, but during this absolute refractory period, that can happen during this relative refractory period. So again, our action potential is flowing from this axon hillock down the length of the axon to the presynaptic terminal area. And this axon hillock is where this action potential is generated. That's where it actually begins. And one of the things that we know is that the action potential, again, is always going in this direction that we see here, right? And what we know is that at these spots right here, this is called the node of Ranvier, the action potential is being regenerated, and that's going to help it to be pushed down the length of this axon down to the terminal endings that you see here. So The action potential does not directly travel down the axon. It's being regenerated at these different points, these nodes of Ranvier that you see here. So again, take a look. Here's a stimulus. Here's the change that's occurring in terms of these channels that are opening up. Potassium now is flowing out. Sodium is flowing in. Again, here's our stimulus. And again, if you take a look at the direction of the action potential, this is occurring all the way down that membrane. And the action potential flows down the axon. It's depolarizing adjacent areas of the membrane as we go along, again, from left to right here. So the myelin sheath, you see right here, that covers the axon, and these little gaps that you see right here called the nodes of Ranvier. These nodes that you see right here, these interruptions, if you will, these are unmyelinated sections. And there's very enriched ion channels that are in each one of these nodes that you see here. And because they are so enriched, what it's causing is what some refer to as a jumping or excitation, which is going to allow these positively charged ions to be pushed even quicker down the length of the axon. So this is an important process, and it is one that is referred to as saltatory conduction. And what you can see here are these enriched ion channels that you see right here, and this jumping process of the action potential from node to node, okay, that you see right here. Again, this is going to provide for very rapid conducting of the impulse, and it's also going to conserve energy in the cell. Interestingly, certain types of disorders like multiple sclerosis, for example, MS, that's a disease in which those myelin sheaths have been destroyed, and it's associated with very poor muscle coordination. So again, this myelin sheath and what's happening at those nodes is incredibly important. The saltatory conduction is important for the action potential. So that serves as again, as a basic understanding of what is happening in terms of the action potential. And we'll now begin to take a look at neurotransmitters in the brain. But again, just by way of a little bit of review, here's our key concepts that you need to be familiar with: all or nothing firing, changes in the permeability of the membrane, changes in axon polarity, and these refractory periods. I think that those are key concepts, okay, in terms of understanding the action potential. So we'll now begin to take a look at neurotransmitters.